research work in the past 17 years in Nigerian University as a researcher. And I'll introduce the topic, discuss Nigerian biodiversity, drug discovery from natural products, the challenges and prospects, then some other research I did apart from drug discovery from natural products. Then I'll be concluding and discussing my future research directions. Can we still see the screen, please?
Yes, yes.
Yes, yes, we are seeing.
Our understanding of the ideas of venom versus antidote and antigen against antibody has taught us that Mother Nature offers remedies for all illnesses. Drug research and biotechnology have evolved greatly in recent years, and biodiversity has become a key source of possible novel medications and treatments. Researchers can now uncover compounds that are useful against particular diseases and develop new therapies and medicines by analyzing the natural molecules produced by different species. This is a priceless source of potential remedies that can be used to improve the health of humans. Well, despite the enhanced health system we have in Africa, we found that in the 21st century, infectious diseases have become a major global health threat, particularly in Africa, of course, where they contribute to health inequality among Africans. Despite efforts to improve health systems and increase access to treatment and vaccination, infectious diseases continue to affect the African population. Diarrhea, malaria, and severe respiratory infections are the most common infectious diseases in Nigeria, and they are the main causes of preventable deaths. To minimize the burden of infectious diseases in Nigeria, a better monitoring and data collection system, better access to medical care, and more comprehensive prevention measures are needed. Cooperation between government agencies, health professionals, communities, and financing of new vaccines and treatments is essential to address the problem. Ethnomedicine, or using traditional knowledge and practices to prevent and treat sickness, has a long history in Nigeria. The use of medicinal plants, a significant source of novel medication, is the foundation of ethnomedical practice in Nigeria. Many traditional healers use a combination of herbal remedies to treat a wide range of illnesses. Prayer and meditation are two basic spiritual practices that aid in the healing process. Nigerian ethnomedicine has changed over time due to adjustments to the healthcare system and advances in technology. Traditional healers, however, still have been a major contributor to the healthcare system, but there have been a shift towards modern medicine due to the need for evidence-based treatment and standardization and regulation. Traditional healers, though, still play a vital role in rural areas with limited access to modern medicine. These are some examples of plants used in the treatment of typhoid fever, as published in one of my research back in 2010. These plants, various parts of the plant are used, from bark Either the bark is boiled in water or the leaves boiled in water and drunk over a period of time. Different parts are used. And apart from treating typhoid fever, these plants are also used in treating other forms of ailments. And this is what the traditional medical practice or ethnomedicine is. And what we try to do is now to bridge the gap between traditional medical practice and modern medicine by finding pharmacological validity for the practice in our ethnomedicine. Nigeria is a country with incredible biodiversity spread across its vast landscape. Nigeria's rich biodiversity includes plants, animals, and ecosystem. The plant and animal life is quite much, and we have thousands of plant species, hundreds of birds, mammals, reptile species, and so on. And for that, Nigeria ranks 11th in Africa for biodiversity. And we boast of one of the 25 global biodiversity hotspots. And this includes our forests, jungles, wetland, and mangrove forests. These areas are home to endangered species such as gorillas and chimpanzees. Additionally, the savanna grasslands are home to large mammals like elephants and large wild cats. Nigeria's rich biodiversity provides numerous benefits to the environment and the economy, including serving as food source for ecotourism, job creation, and raw materials for agricultural and pharmaceutical industries. However, Nigeria faces significant threats to its biodiversity from population growth, anthropogenic activities, and climate change. Implementing effective conservation strategies involving local communities is essential for sustainable resource use and management. There's a growing interest in natural products and traditional medicine globally, and Nigeria has a potential to become a significant player. Our biodiversity, including medicinal plants, microorganisms, and marine organisms, can place us on the global map in this respect. And with the renewed interest to medicinal plants and plant-based medicine, it puts Nigeria in a vantage position. Several medications have been produced from Nigerian medicinal plants, but standardization of production and handling method is needed to ensure the safety and the quality of this medicine. And this can be achieved through investment in pharmacological biochemistry research to carry out plant-to-pill conversion. Nigeria also have a very large population of researchers, and our biological diversity provides a unique opportunity to explore the potential of plant-to-pill conversion research. Nigeria's biodiversity is, however, underexplored and understudied, even with many plant species potentially containing novel chemical compounds with potential therapeutic applications. The opportunities for Nigeria to leverage on its vast natural resources to develop plant-based medicine that can compete in the global market. Natural products are small bioactive compounds produced by various living organisms. These can be whole organisms, part of an organism, extracts, or pure compounds isolated from plants, animals, or microorganisms. And they can include things like alkaloids, coumarin, flavonoids, lignans, steroids, terpenoids, and so on. Natural products often serve as pesticides, fragrances, and medicines. However, in practical usage, the term natural product typically refers to secondary metabolites, which are organic compounds that have biological activity, but do not serve a primary function in the growth, development, or reproduction of an organism. Secondary metabolites are primarily produced by microorganisms and function as antibacterial defense against other germs. Plant secondary metabolites aid in stress tolerance and cellular defense. In animals, they may function as hormones, neurotransmitters, and immunological stimulants. These compounds vary in structure and biosynthesis and are classified based on their metabolic pathways. And this offers Nigeria potential to develop plant-based medicine from natural products along this line. So in discussing drug discovery is the process of identifying new candidate medication. We should state at this point that drug discovery is not the same thing as drug development. My research is basically on drug discovery. In recent years, the research into novel drugs from natural sources has gained considerable attention because such compounds are often endowed with high bioactivity, as well as long tradition of safe use, resulting in a reduced time and cost for clinical development. Drug discovery is the process of identifying new candidate medication. Over the years, drugs have been discovered through various methods, such as identifying active ingredients in traditional remedies, through accidental discovery, or selecting synthetic small molecules, natural products, or extract from intact cells and whole organisms in chemical libraries. This process of drug discovery is known as classical pharmacology, and it allows researchers to identify compounds that can be used as medication and developing effective treatment for a variety of diseases and conditions. This is a schematic representation of the process of drug discovery from natural products. When you have an extract, you want to purify the extract through fractionation. And once purification is done, you have a pure compound. The next thing to do is to do what we call structure elucidation, so as to know the structure. If you have a structure for the compound, then you want to get a target, a biological target that the compound can have activity on. These biological targets could either be proteins, they are usually proteins, enzymes or proteins. And they are such that they are parts of the disease cycle in such a way that if it is modulated, it can change the outcome of a disease. So if you discover such a protein or an enzyme that such a compound binds to and modulates, then you have discovered a target and you validate such targets. Once you have done that, you have a lead identification, and the next thing is to want to optimize the lead by carrying out certain tests to ensure that the lead is safe and can be, further research can be carried out on it so that a new candidate drug can be produced from it. So historically, the discovery of drugs was made by identification of active ingredients from traditional remedies. by serendipitous discovery. Substances, whether crude extracts or pure chemicals, have been tested for biological activity without knowledge of the biological targets. Only after the identification of an active substance can efforts then be made to identify the target. This is what used to be done in classical drug discovery. The discovery process includes the early phases of research, which are designed to identify an investigational drug and perform initial tests in the lab. The initial phase of natural product-based drug discovery involves extensive bioprospecting to identify promising plant species harboring desired therapeutic molecules. Modern extraction and analytical techniques offer unprecedented access to this vast biodiverse reservoir, aiding in the efficient isolation and characterization of lead compounds. However, optimizing these leads for clinical viability presents a significant hurdle. The stringent demands of potency, selectivity, safety, and pharmacokinetics often lead to high failure rates in clinical trials. The inherent complexity of natural products further exacerbates this challenge, and this necessitates innovative strategies for structure-based modification. Fortunately, recent advances have revitalized this field, and this development have fueled renewed interest in natural products, particularly for their potential to combat emerging disease threats and antibiotic resistance. The first step in drug discovery from natural products is to isolate and characterize natural products from its biological source. Excuse me. Natural product isolation and characterization are therefore critical steps in drug discovery process. And in my research, we analyzed some medicinal plants in Nigeria for the presence of certain bioactive components in the crude extracts. And these were published Musa et al. 2011, Usman et al. 2021, and so on. And we discovered a lot of bioactive components in these plants. Also, in trying to get new treatment remedies or new treatments from traditional medicine, you have to validate what is done in the ethnomedical practice. The choice of their solvent of extraction, we tried to validate that in the lab, and we were able to provide pharmacological basis for the choice of solvent of extraction used in traditional medicine. Sometimes they use water, sometimes they use alcohol, and we're able to do a validation of some of these choices. Then the temperature of extraction too was also validated in the lab. Sometimes in traditional medicine practice, some of these extracts are boiled. At other times, they are macerated over a period of time. So we try to follow what is done in traditional medicine to see if there was a pharmacological basis for what they did, and we're able to validate some of these things, although some we could not validate. And in isolating and characterizing compound from a natural product, we're able to isolate certain compound from two plants used in the treatment of typhoid fever in Nigeria. And these are examples of some of the compounds that we isolated from these two plants, Eucalyptus camaldulensis and Ficus capensis. We investigated the antibacterial activities of these plants to validate the ethnomedical practice. My research was mainly on typhoid fever, and I did a lot of work on typhoid fever to see what is used in traditional medical practice to treat typhoid fever, find out if we can get pharmacological validation for it, and then step the research further into discovering probably new treatment regimens for typhoid fever, which still remains a public health issue in Nigeria and other African countries. And we're able to demonstrate that some of these plants actually have anti-typhoid activities. The step towards purification is from crude extract, you do bio-guided fractionation. You fractionate, keep fractionating until you are able to get pure compounds that are responsible for the activity of the plants against the bacteria or the microorganism. And in some cases, we discovered that fractionation leads to reduced activity, whereas in other cases, fractionation increases the activity, which should have been the best way to go, or the right order. Fractionation, as you fractionate, the quantity of the active principle increases, and so the activity should also increase. But we have seen some cases where fractionation leads to reduced activity. And this could be as a result of the fact that probably several active principles are responsible synergistically for the effect we see. And when they are fractionated into different fractions, the activity reduces. They are no longer in the same solution to be able to carry out the synergistic effect. We discovered also that some of the plants that are used in combination for the treatment of typhoid fever in our ethnomedicines, and there were no pharmacological basis for this practice, as the individual plants have higher activity than when you combine them. And some of these activities are actually comparable to standard antibiotics. This is an example of a research where we saw that fractionation led to reduced activity of these plants, extract of Morinda lucida against Salmonella typhi. The crude extracts had activity which was comparable with gentamicin, much better than amoxicillin and chloramphenicol, but the fractions had lower activity. This shows that perhaps in different fractions are compounds that may synergistically act to provide the increased activity we see in the crude extract. We also screened for antioxidant activities of our extract, and we saw that a lot of our extracts have very high antioxidant activity compared to standards such as gallic acid and ascorbic acid. During COVID-19, we carried out a preliminary study into certain vegetables or commonly eaten foods in Nigeria. And the hypothesis behind this work is that even though it was expected that there will be a lot of casualties from COVID in Africa, we didn't see that. Even with our very poor health facilities, we didn't see the kind of casualty that we're expecting. So we're thinking that one of the things that have given us protection against COVID could be in the food we eat. Several theories were put out, and we were able to disprove a lot of them. Some talked about the fact that there was high sunlight in sub-Saharan Africa, and there is a heat that was destroying the organism, but this is not true, and several other things. So we decided to look at food, and we selected some plant samples from commonly eaten food we use. In my lab, I do not have a BSL-3 facility, so I decided to use a model organism to represent the virus. I used the bacteriophage MS2. And we did this research such that we wanted to test the activity of this plant extract in physiological condition as well as in environmental condition. In physiological condition, we try to mimic what happens within the body, and we use a buffer for that. Then in the environmental condition, we used water. And we saw that there was activity, especially by two plants, against this, our model virus. There was high activity. And we are trying to look for funds to be able to carry out, to be able to proceed with this research, because what we have seen here is interesting, and we want to follow it to the logical conclusion. And perhaps this could help us in preparing for the next emerging infectious disease. And talking about plants, one of the thing people usually say here is that anything that is natural is safe. But we know that even for natural things, we have the good, the bad, and the ugly from the natural things. So plants can also be very, very toxic, because medicinal properties in the plant are due to its chemical constituents. And some of these chemical constituents can be very dangerous if consumed. So we carried out a lot of toxicity studies on our plants that we used, that we're studying in the lab, that is used in the treatment of typhoid fever. And we discovered that the plants are actually safe for consumption, and most of them had LD50 above 5,000 milligram per kilogram body weight. And the result of other toxicity studies we carried out showed that they were actually safe for consumption. Having seen that our plants have pharmacological activity and they are safe, we now try to look at the possible mechanism of action by which these plants carry out their activity. And from our studies on Eucalyptus camaldulensis and Ficus capensis, they may carry out the antibacterial activity by membrane stabilization, leukocyte mobilization, and inhibition of prostaglandin synthesis, as well as phospholipase A2 enzyme. Excuse me. For membrane stabilization, we know that membrane stabilization actually protects bacterial cells from hypotonicity-induced damage. However, membrane stabilization can also make the cell become more rigid, prevent the release of intracellular content, and make it difficult for bacteria to take up nutrients necessary for growth and reproduction. Bacterial growth is therefore inhibited because of the prevention of the uptake of nutrients and the release of intracellular content, which can damage the cell and lead to cell death. In this table, We see that extract of Ficus capensis and Eucalyptus camaldulensis inhibited hypotonicity-induced hemolysis in a concentration-dependent manner. So we hypothesize that one of the ways that these plants could be carrying out their activity may be through membrane stabilization. Leukocytes are white blood cells which fight infection by attacking and killing bacteria. Leukocyte mobilization involves moving leukocytes from the bloodstream to the infection site. Once leukocytes are mobilized, they begin to attack and kill bacteria by utilizing phagocytosis, antimicrobial release, and antibody production. This table shows that compared to animals in the control, which received just normal saline, the total leukocyte count of the animals treated with extract of Ficus capensis and Eucalyptus camaldulensis increased. Thereafter, it decreased with increasing concentration of extract treatment. There are gradual decrease in the percentage of neutrophil mobilized with increasing concentration of extract used in the treatment of the animals. However, there was an increase in the percentage of lymphocytes mobilized as the concentration of extract used for the treatment of the animal increased. This result shows that extracts are capable of mobilizing leukocyte, which is an acute inflammatory process that aids in antibiotic activity, but also capable of preventing chronic inflammatory process by increasing the concentration of the extract. In the case of prostaglandin, prostaglandin are a group of lipids that play a role in a variety of bodily functions, including inflammation, pain, and fever. They are also involved in the immune response to infection. Inhibition of prostaglandin synthesis can therefore aid antibacterial action in a number of ways. First, it can help reduce inflammation, which can make it easier for antibiotics to reach the bacteria. Secondly, it can help reduce pain, and it can also reduce fever, which can also make it easier for antibiotics to work. This table shows that the extract of Ficus capensis and Eucalyptus camaldulensis inhibited prostaglandin synthesis. Excuse me. Phospholipase A2 activity. Phospholipase A2 is an enzyme that breaks down phospholipids, which are important components of the bacterial cell membrane. Inhibition of phospholipase A2 can cause antibacterial action by membrane disruption, cell wall synthesis inhibition, and DNA damage, leading to the cell death. This table shows that the extract of Ficus capensis and Eucalyptus camaldulensis induce significant concentration-dependent inhibition of the activity of phospholipase A2 when compared to control. So what are the challenges in drug discovery from natural products? The challenges are much, and they include funding. Funding is a very, very big challenge to drug discovery from natural products, especially in Nigeria. Another thing is the challenge of time. After identifying a hit in an extract, bioassay-guided fractionation is required to determine the active component. This time-consuming technique is not necessarily consistent with the current regime of bleed screening programs. Excuse me. In which assay support is only available for limited period. And this is one of the reasons why Big Pharma actually left drug discovery from plant or natural products. Time was an important factor for them. Another factor is technical barrier. There are technical barriers for screening, isolation, and characterization of natural products. Because natural products are often a complex mixture of compounds, and it can be very difficult to isolate the active compounds responsible for a particular biological activity. This can be time-consuming and expensive process in isolating the active compound. These technical barriers could also include complexity. Natural products are often complex molecules, which make the isolation and characterization difficult. There's limited supply. Once you get a lead from a plant, what are you going to do? Are you going to cut down all the trees or the plants to be able to get more supply? So the supply from natural products is actually limited, and it's one technical barrier. We have several other technical barriers. We have barrier of the problem of a challenge of biodiversity loss. This is caused by deforestation, urbanization, and other human activity. As a result, the number of plant and animal species available for drug development is decreasing daily. There are regulatory hurdles to overcome in carrying out drug discovery from natural products. And also, countries are increasingly prioritizing biodiversity conservation, demanding prior authorization for natural product exploitation. This issue of intellectual property brings another barrier and challenge in drug discovery from natural products. And when you come to Nigeria, the problem even increases because of lack of infrastructure. Nigeria lacks the infrastructure required to enable drug discovery, notably well-equipped laboratories and competent personnel to assess high-quality analytical equipment are lacking. Therefore, there's need for increased funding of research in this sector. Now, this leads to the poor funding. The Nigerian government's lack of adequate research funding severely limits the resources available for drug development from natural products. We also have limited documentation and knowledge of our vast biodiversity. There is inadequate documentation, and because of these, many plant species used in traditional remedies have not been scientifically studied or catalogued. We also have problem of quality control and standardization. Even though we have a lot of plant-based medicine in Nigerian markets, but there's issue with standardization and quality. So those are some of the issues with drug discovery from natural products in Nigeria. Then one other very key issue is inadequate collaboration and networking. We find out that in Nigeria, we see pockets of researchers repeating the same research in different laboratories instead of collaborating and networking so that we can advance our work instead of repeating the same thing. So collaboration and networking is one very key challenge in Nigeria. Well, the prospect of drug discovery from natural products is increasing, and that is a good thing. If Newman and Craig 2020 reviewed the sources of new approved drugs for treatment of human diseases from 1981 to 2019. And this as shown in this figure here. The utilization of natural products is increasing in the number of drugs approved within this period. And this is a source of good news. So what are the prospects for drug discovery from natural products? There are a lot of technologies out now that now gives drug discovery from natural products good prospects. Natural products have enormous promise for drug discovery in the future, and several approaches and breakthroughs have paved the way for fascinating new trends in this area. Pure natural products may now be obtained in considerably greater quantities than before, all thanks to these new techniques. Researchers are now focusing on reducing the process from screening to lead identification. High throughput screening is one of the techniques used. Another is combinatorial chemistry and combinatorial biosynthesis. And this approach potentially can help in identification of natural products for drug production. In combinatorial biosynthesis, genes from several species are combined to produce new natural compounds with novel biological activities. In combinatorial chemistry, chemical building blocks are combined to produce new molecules with novel structures and biological properties. These methods increase the chemical space available to be explored, while also producing molecules that are possible for effective and less toxic than the natural product prototypes. Phenotypic screening continues to be a key strategy in the development of drugs from natural products, in contrast to target-based methods that have been routinely employed in the pharmaceutical industry now. Therapeutic drugs, target-based screening. Strategies include polypharmacology, proteins known as drug transporters, immunomodulation, and computer-aided drug design, which I'll be discussing later. Yes, we also try to use computer-aided drug design to look at these compounds that were isolated from 2 plants in Nigeria. And we try to dock it against 2 proteins that are disease targets for typhoid fever, the ROE protein and the PAN-C protein. And we found some interesting discoveries. The compounds were docked against ROE and PAN-C, which are protein targets for treatment of typhoid fever, to provide further insight into their therapeutic potential for the development of novel anti-typhoid treatment. The docking process involved a prediction of the ligand conformation, as well as its position and orientation within the binding site, and the assessment of the binding affinity, which is the docking score. In docking the protein, in docking, the protein is rigid and only the ligand can move, while molecular mechanics generalized bond surface area, MMGBSA methods, account for protein flexibility and solvation to mimic the real system. Thus, MMGBSA binding affinity supersedes the docking score. And we discovered that in this table, for instance, this compound has the best docking score as well as the best MMGBA score. And this is the 3D diagram of the interaction of this compound with ROE protein. And for this table, we see that there are two different compounds with the best docking and MMGBSA score. And like we said before, that the MMGBSA score supersedes. So for the last compound here is actually the best in interacting with this PAN-C protein. And this is the 3D interaction of the ligand and the protein. So these are some of the research we've done in drug discovery from Nigerian natural products. And from our computer-aided drug design research, we're able to find that apart from, because we started work with trying to look for novel treatments for typhoid fever, we discovered that this plant also contains a lot of other phytocomponents, chemicals, that potentially could have effect against several other diseases, from the fishing we did using a virtual screening of the compounds we did. We found out that they could have potentially significant effect on other disease processes. And so that is another next line of action for us to carry in our lab to find out how these other compounds are able to affect the other disease that in silico screening shows that they're capable of having effect on. Apart from that, we also did some research on drug design and delivery. We did researches in biotechnology and molecular biology, clinical biochemistry, nutrition, toxicology, and so on. And all these things we try to look at Nigerian biodiversity and how it can improve our health and also can lead to sustainable development in Nigeria. And some of the things we have contributed to the body of knowledge is that we have explored the ethnomedical claims and provided pharmacological validations or otherwise for some of the ethnomedical practices in Nigeria. We have contributed to the library of plant compounds by characterizing pure compounds from Nigerian medicinal plants. We have contributed to the understanding of therapeutic potentials of natural compounds from Nigerian biodiversity and paved the way for further investigation. We have developed prototypes for new drugs that may address some of these African health challenges. We have revealed that the antibacterial effect of some of the medicinal plants are mediated through membrane stabilization, leukocyte mobilization, and inhibition of prostaglandin synthesis, as well as phospholipase A2 activity. We have also potentially developed an alternative plant-based eco-friendly larvicide against malarial disease. In drug design and delivery innovation, we're able to develop pharmaceutical grade starch from underutilized food and non-food starches in Nigeria. There is a non-food starch in Nigeria that we're able to enzymatically modify, and the products we got compares favorably with pharmaceutical grade starch. Unfortunately for us in Nigeria, our pharmaceutical industry still continue to import all their pharmaceutical grade starch. We don't produce it locally here. So by these developments, if we improve on it, this is one of the things that can lead to sustainable development in our economy, because it will reduce the number of foreign exchange we spend on importing some of these things, and we'll now be able to spend money on our own economy. Also, there are some food starches that we also develop into pharmaceutical grade starch, but there is no fear of competition with food here, because even though they are food starch, there are some variety of rice developed in Nigeria, which farmers have stopped farming because when you cook the rice, it loses its consistency. So farmers have stopped planting such type of rice. So we took those rice and we decided to enzymatically modify it, and we're able to get very good pharmaceutical grade starch. So it can now encourage farmers to begin to plant that variety of rice again, and it will not compete with food itself, even though it's a food starch. We have contributed to rational drug design effort by predicting the 3D structure of novel therapeutics against multi-drug resistant strain of Salmonella typhi. We have also produced pharmaceutical grade collagen from non-conventional sources, such as fish waste, and also produced biofilms from enzymatically modified cocoa starch. All these things I'm talking about are from our biodiversity. We have also chemically and enzymatically modified cassava starch for various industrial applications. We have extracted urease from sprouted melon seeds for laboratory and industrial applications. All these are things we have done from our biodiversity, that if there is investment in this area, it will lead to improved health and sustainable economic development. We have confirmed the inefficacy of the present standard diagnostic procedure for typhoid fever in Nigeria, and we have proposed molecular-based PCR techniques for routine diagnosis, because we have seen a lot of times the things that lead to resistant to antibiotics is abuse of antibiotics. And when drugs are wrongly prescribed for the wrong disease, it could lead to such resistance. And a lot of times, people have been diagnosed to have typhoid fever in Nigeria without them having typhoid fever. And this is one of the things our research has cleared. We have also established the prevalence of resistant genes such as black-tem and cat-p genes in Salmonella isolate in Nigeria, and hence the resistance to routinely used antibiotics in the treatment of typhoid fever. We are currently carrying out a funded research on molecular characterization of Salmonella typhi in Nigeria. And by the end of that research, we still have more results to give to the public on that. We have contributed to neonatal care by identifying risk factors and biomarkers for neonatal instability from cord blood. Cord blood is usually discovered at birth in Nigeria, but we have identified some biomarkers in the cord blood that can identify risk factors in a child, and this has contributed to neonatal health in Nigeria. And we have also established that from cord blood, we can use some biochemical and hematological indices to predict transplacental or congenital malaria in neonates. These are some of our contributions. And we have contributions in other areas. In Nigeria, there are no labelings for GM food. And we have established GM fingerprints in maize and soybeans in Nigeria. Those sold in the markets, the products, and even in feeds, animal feeds. So this is raising awareness on the need to enforce compliance with GMO labeling regulation by authorities in Nigeria. These are some of our contributions we've had over the years from our research in the last 17 to 18 years. And in the future, from now, there are certain other things we want to do as part of our contribution to science from now on. And one is to establish a biodiversity park. By creating a biodiversity park, I would love to carry out biodiversity conservation of significant Nigerian medicinal plants. In this line of research, the most significant medicinal plant species in Nigeria will be identified, and a park will be established to safeguard them from risk of deforestation, increased agricultural production, and other human activities. A park of this kind will provide a protected area where these plants may thrive and their genetic diversity can be maintained. In addition to assisting in preservation of these significant medicinal plants for future generations, the establishment of a biodiversity park will encourage ecotourism in Nigeria and serve as a platform for a scholarly investigation into their characteristics and prospective applications. Another thing I would like to do is creation of a centralized database of natural products isolated from Nigerian medicinal plants. Researchers, pharmaceutical companies, herbal medicine practitioners will benefit from the creation of such a database of natural products isolated from Nigerian medicinal plants. This database will allow researchers to save time and money by avoiding the need to isolate, characterize, and synthesize substances that have already been studied. It could also speed up the drug discovery process in Nigeria, benefiting patients in need of new treatments and resulting in the discovery of new drugs and therapies for various diseases. Additionally, the application of cutting-edge technologies like machine learning and artificial intelligence may improve the precision and efficacy of drug discovery, cutting down on the expense and duration of research and development. This could result in more medications with Nigerian medicinal plant origin being approved, helping to grow the Nigerian pharmaceutical industry and providing employment opportunities for local scientists and researchers. The third and final thing I would like to do is molecular epidemiology of important diseases in Nigeria. This research will involve the application of molecular biology techniques to study the genetic and environmental factors that contribute to the spread and severity of specific diseases in Nigeria. This research will help identify the most vulnerable populations and develop targeted interventions to prevent or treat these diseases. It could lead to the discovery of new drugs, new drug targets, and development of novel therapies for these diseases, as well as inform public health policies and guidelines to reduce the burden of these diseases on society. The knowledge can also aid in vaccine development and improve treatment strategies for individuals affected by these diseases. Also, understanding the genetic and environmental factors that contribute to the development of diseases can help identify high-risk populations and implement preventive measures. This knowledge can then inform policy decisions surrounding healthcare and genetic testing. My recommendation, therefore, is to expedite drug discovery. African researchers should work together and share their expertise and resources. By doing so, they can explore the pharmacological potential of Nigerian natural resources, identify potential lead compounds, and bridge the gap between traditional medicine and modern pharmacology. Collaboration should also extend to overseas research groups, such as the eminent researchers we have in LSI-NG, to include in the sharing of these findings. Nigerian and African university management should invest in drug discovery, research infrastructure, and facilities to attract and retain skilled researchers and students, encourage innovation, and promote engagement with the wider community. And this recommendation is going to university managers across Africa and Nigeria in particular. On the part of the government, they should increase funding, create policies, and provide incentives to encourage drug discovery research. Government should also establish regulatory framework to ensure the safe and ethical exploration of natural resources for medicinal use while promoting biodiversity preservation for drug discovery. To the private corporate organization, collaboration with academic and research institutions. Excuse me, let me get water. My throat is dry. Let me get water, please. Sorry about that.
No problem.
Collaboration between private corporate organizations and academic and research institutions can optimize research outcomes by providing funding, partnering with researchers and universities, and investing in drug discovery. Additionally, private companies should prioritize biodiversity conservation, support sustainable practices, and facilitate the responsible management of natural resources, while also creating opportunities for the commercialization of successful research results. And to funders, grant agencies, they should encourage funding for pharmacological biochemistry research in Nigeria and Africa, focusing on natural resources. Funding agencies should support collaboration and multidisciplinary research among researchers and industrial partners, as well as promote outreach and engagement initiatives for result dissemination. In conclusion, this webinar has highlighted the enormous potentials of Nigerian rich biodiversity for transformative global impact on health and sustainable development. My 18-year research uncovered natural treasures, advanced drug discovery, and pioneered innovation in pharmacological biochemistry. By utilizing indigenous flora to combat antimicrobial resistance and advocating for biodiversity conservation. Our efforts have significant implications. The combination of traditional knowledge, cutting-edge technology, and collaborative partnership highlights Nigeria's critical role in shaping a healthier and more sustainable future. And in putting my thoughts together, how we can leverage on biodiversity to improve our health and to have sustained development is that if we invest in drug discovery from Nigerian biodiversity, we will be able to produce newer medications, perhaps cheaper for us in the Nigerian market, and this will lead to increased healthcare delivery. If you have a healthier society will mean that the society will be able to be more economically viable, as people will be able to work and improve the economy. In addition to that, if we develop our biodiversity, it could place us in a situation where we now spend less forex on importing drugs, and we develop our own pharmaceutical industry, and this will lead to sustained development in Nigeria. In my years of research, I have to acknowledge my research mentors that have helped me over the years. My colleagues from Novartis, from TDRC, from LSINJ. Of course, this is not a comprehensive list, but this is just representative of LSINJ. My family, my parents and siblings, my children, and of course, my wife. Thank you for listening.
Oh my God. What a powerful program. God bless you.
Thank you very much, Dixon. It's great talk. Does anybody else want to ask questions before I do?
Oh, that was one of the greatest lectures on medicine I ever heard. God bless you, sir.
Thank you.
Thank you so much.
Thank you so much, Professor. Thank you so much.
Thank you, David.
Okay, Ferris, you say we should ask questions?
Well, yeah, go ahead, and whoever wants to ask questions, I have them too. It'd be great. It was a great talk.
Okay. Yeah, my name is Ibinoma Andrew Aigboye. I'm from Nigeria. Thank you so much, Professor Dixon, for this elaborate presentation and your contribution to science. I really appreciate your research contribution. Thank you, sir.
Thank you so much.
Nigeria has over 250 ethnic groups, and many of these groups have their different belief and traditional approach when it comes to administering any therapeutic agent they believe. I mean, probably traditional medicines. And I wanted to ask, should we believe that this diversity in approach can contribute directly or indirectly to a possible disparity in antimicrobial profile to conventional antibiotics, or it has no way of altering antimicrobial profile, their response to conventional antibiotics? That